Hey everybody, today we're debating human origins, and we're starting right now. Ladies and gentlemen, thrilled to have you here for another epic debate. This is going to be a fun one, folks, as today it's going to be a debate on human evolution, or Adam, Eve, and Noah of the Ark. So this is going to be a lot of fun. Want to let you know we have two very experienced debaters with us today. These guys are passionate about what they do, and that's what makes it fun. So want to let you know, though, if it's your first time here, consider hitting that subscribe button, as we've got a lot more debates coming up. We're very excited about, for example, is the Illuminati real? That will be coming up on Saturday as an example. So it should be a juicy one featuring our own Tom Jump, who we love very much. Terrific guy. And want to let you know as well, whether you be Christian, atheist, if you take the creation side or the evolution side, no matter what it is, we do want you to feel welcome no matter what walk of life you're from. We are a neutral channel, so we don't have any positions ourselves. We just leave that to the debaters. And we want to thank them very much for being here. It's an honor to have both RJ and Standing for Truth with us tonight, as this is going to be a lot of fun. Thanks so much, gentlemen, both of you, for being with us.
Glad to be here. Had a Sunday to kill.
I'm excited, James. You know I'm always excited to be here, so thanks so much.
The pleasure is all mine. And want to let you know, folks, they're linked in the description. So if you're listening and you're like, I like that, I want more, you can hear plenty more at their links that I put in the description for you. So it's going to be a lot of fun. Want to let you know what we're going to do is we're going to have 7-minute opening statements, 45 minutes of open conversation, and then roughly 3-minute closings, followed by Q&A. And with that, want to let you know, you can, if you'd like to ask a question, fire your question into the old live chat. And if you tag me with @ModernDayDebate, that makes it easier for me to be sure I get every question in the list for the Q&A. And super chat is also an option, in which case not only can you make ask a question, but you can make a snarky comment. So for example, towards
Please ask questions.
you can make, it could be about Standing for Truth, his presentation, his appearance. It could be anything you want. So we are very excited. It's going to be a lot of
or
fun.
RJ being an old fart.
These guys are the best. They're terrific. And we want to let you know, though, we're very thankful that these guys have got a good amount of time. So I would say one thing that we've sometimes run into lately is once in a while, to respect the time of the debaters, we have to close early, and we've not gotten to read all the super chats, which I hate, because I think the purpose of super chats is for people to be able to engage. So want to let you know, there's like probably I'd say an 85% to close to 90% chance that we'll read every single super chat tonight, but just want to let you know, once in a blue moon we don't get to read all of them, and that honestly bothers me. So with that, very excited. So we're going to get right into this with gentlemen, I'm so sorry, I'm embarrassed to say I forgot to ask who's going first.
Oh, apparently I am.
You got it. Okay. Well, the floor is
Standing asked me if I'd like to go first, since he went first the other times, and I said, yeah, I'm okay with that.
You got it. So I'll pull up the timer, and thanks again, gentlemen, for being here. The floor is all yours, RJ.
Okay. Hopefully will not take as much time as they need. I'm assuming that Standing is going to make his arguments about created heterozygosity and all of the various claims about endogenous retroviruses. We've heard a lot of that before. I'm actually not here to go over the whole structure of human evolutionary theory. You can channel and look at Australopithecines and DNA and all this stuff. There's got websites up the yin yang on that. It just is that we know that there is an evolutionary data set out there, and Standing for Truth won't like any of that. And yeah, we get that. But what I am most interested in is finding out more about Standing for Truth's creation model. Particularly in relation to the peopling of the New World. We'll want to find out, were there any people here prior to the flood? That's that period between Adam and Eve and Noah's Ark. And if not, then how did the people get here that got here? Presumably, they are from the 8 people that were on the Ark, Noah and the kids. And therefore, we have to figure out what's going on there. They have to build up a population. How big of a population? Because remember, they're populating the whole planet, supposedly. So which ones of those are the ones that are going across to the Americas? How do they do that? When do they do that? What kind of food crops are they carrying with us? We know that Native American cultures have maize, aka corn. They've got potatoes down in the Andes. They've got chocolate that comes from the New World. We've got tomatoes. There's a whole mass of endemic plants that are not known anywhere else in the New World, in the Old World, and vice versa. The wheat, rice, and other material that's known in the Old World, barley and all that, not known in the New World. So how come the differentiation of what was taken and when? Farm animals, did they have any of those? Had they domesticated like horses or cattle or anything else? Horses, and beyond that, we can bring up maybe camels. We can talk about mammoths and all sorts of things that relate to what Native Americans might or might not have been doing. And does that chronology fit at all in the structure of the flood model? It's an important issue. We're in the United States and the Americas. We're not in Europe. We're not living in someplace else. We're here. And so it would seem the most natural thing in the world to figure out what's going on with the peopling of our hemisphere. So that covers pretty much the basic points that I want to get across. And I'm only not even 3 minutes in, and the rest of that can go to our discussion.
You got it. Thanks so much, RJ. We will kick it over to Standing for Truth for his opening statement. Standing for Truth, thanks for being here as well. The floor is all yours.
Awesome, James. Just 1 second. Let me get myself set up here. Thanks so much for that opening statement there, RJ. Thanks for doing this debate and setting it up, James. Can you see my screen?
Oh, yeah.
Okay. Let me get my timer set. I'll make this short and sweet. I'll set it for 7 minutes. And here we go. Okay, so what should we expect to find if Adam and Eve were true? It should be obvious to anybody that what we should expect to find is evidence for one female ancestor of all people on the planet, as well as evidence for one male ancestor of all people on the planet. Well, genetic data confirms these expectations. He did talk about the Australopithecines very briefly there. Well, they share most of their features with living apes today. They were only around 3 or 4 feet tall. Yes, they did have some uniqueness in their morphology, but this means the best conclusion regarding Lucy and the Australopithecines is that these apes were really nothing more than just an extinct ape genus. That's really only the only thing I heard about the evolution side. But evolutionists will argue against Adam and Eve by pointing to current human genetic diversity. There are too many DNA differences to explain from just 2 people over 6,000 years. This would be true only if mutations were the source for all variety. Time is not required for the diversity today because diversity can be designed and built into Adam and Eve. RJ here is faced with a serious challenge tonight. This challenge involves pedigree-based human mitochondrial DNA mutation rate studies. The reason for this is because pedigree-based studies in the empirical method rejects deep time evolution and universal common ancestry. Evolutionists look to time dependency to explain away the data. Evolutionists would have us believe that mutation rates have been significantly slower in the past. For this explanation to be scientific or even reasonable, James Downard must make testable predictions. If RJ disagrees with the Eve date derived from straightforward mitochondrial DNA coalescence equations, he needs to make testable predictions. When does the molecular clock speed up and slow down? Can RJ here predict when the mutation rate accelerates and when it does not? Or is time dependency a rescue device that evolutionists are forced to invoke in order to explain away the observable data? Maybe RJ can answer that question at the start of the discussion. When we focus on the variation in the Y chromosome in the world today, we see that Y chromosomes have very little variation. This indicates that we came from a common ancestor in the not so distant past. A new research paper here by Nathaniel Jensen confirms that we only observe approximately 4,500 years worth of mutation accumulation in the Y chromosome. Also, subsequent papers, one is seen here, confirm testable predictions on the history of civilization, which is what RJ spent the majority of his opening on. And testable predictions have been confirmed, and it's found in the Y chromosome, genetic signatures, for example. So how does James deal with this? Maybe he can answer it in the discussion portion. How does James explain the mitochondrial DNA and the Y chromosome data that, based on the empirical method, tells us we come not from just a small founding population, but a population of just two, Adam and Eve, 6,000 years ago? We are making predictions on DNA function, mutation rates, speciation rates, and more. What directly records a species ancestry? Well, it's in genetics. It's in our DNA where the question regarding ancestry is answered. The best evidence for human evolution has been overturned. The entire junk DNA paradigm has been overturned. The chromosome 2 fusion has been overturned. Now, these are the lines of evidence you would find if you were to go look into the source data as RJ recommended in his opening. So I'll just refute all those right here and now. So for example, the chromosome 2 fusion, for many reasons, it's been overturned, but most importantly, and I'd like RJ to answer this in the discussion, chimps have very distinctive and exceptionally large satellite sequences at the ends of their chromosomes. Now, where are these large ape-specific satellite sequences in the reputed fusion site? How does James address these problems? ERVs, ALUs, and other classes of retrotransposons, as he went over briefly in his opening, and even pseudogenes are consistent with the created heterozygosity model. I know James loves that word, so. The evidence is clear. These are functional DNA elements important to our genome. Now, the question is, what evidence is agnostic to this debate? Classic textbook examples of universal ancestry include homology, right? That shared structure seen in the biological world. But we know human engineers design in homologous patterns. Across the globe, we see shared designs and even shared blueprints. What about the so-called existence of transitional forms? Mammal-like reptiles, as RJ loves, Archaeopteryx, Tiktaalik, for example. Well, think of a military vehicle that blends the features of both a land vehicle and a vehicle built for the ocean. For example, an amphibious assault vehicle. This would be the perfect example of a transitional form, according to James here, when in fact we know this is not the case. This amphibious assault vehicle was built and designed this way. Nested hierarchies in DNA, anatomy, and physiology are also predicted by creationists based on the design model. Nested hierarchies are products of design. These lines of evidence that James here turns to, typically the ones you'll find in textbooks or the sources that he recommends us look into, to argue against Adam and Eve and a limited ancestry, have far too many explanations that can be invoked, they are agnostic to the debate as a whole. They cannot differentiate between the models. These lines of evidence do not work since both models fit the data. It's a lot like saying the sky is blue or the earth is a sphere. Therefore, creation is wrong. Excuse me, both models can explain the shape of the earth and the blueness of the sky. What does that have to do with the debate? There's a whole lot of explanations that can explain the data. I have just over a minute here. I'll ask a few questions. Just 2 lines of evidence that can differentiate between the models, though, and I hope James here is not afraid to discuss, has to do with molecular clocks, as I described above, and DNA function. We have preliminary evidence for genome-wide functionality. This is a direct prediction of the created heterozygosity model. Can James refute this? How does Downard explain the incredible dissimilarity between TRIMP and human Y chromosomes? How does Downard explain the low genetic diversity in human beings? Very importantly, actually, we know that ERVs and other classes of retrotransposons, including ALUs that RJ loves so much, accomplish many crucial functions in regulating gene expression, differentiation, development, and even cell stress responses. I showed about 6 or 7 papers confirming this. How does RJ fit this into his evolutionary story? I've got a few more questions here, but I'm coming up to the 7-minute mark. I got 10 seconds left. Thanks so much. I'm looking forward to the discussion portion. James, we'll let you kick it off. What kind of testable predictions can you make?
Well, first off, I'll take a point to note that no, the chromosome 2 fusion has not been refuted, although obviously people like Tompkins and Jensen insist that that's the case. Do your own research, follow up on all that material, find out all the problems. Go try source checking them for the detailed elements of it.
Mitochondrial... RJ, I don't want you slapping and running. So the one question, I mean, there are questions I could ask you, right? Where's the cryptic centromere? Why is the area so degenerate? But the one question that I found to be most important that I acknowledged in my opening thing regarding the ape-specific satellite DNA that is found in all apes and should
You got any geneticists that's agreed with you on that other than the creationists?
I did show a paper here from the secular literature that goes over this in great detail. Of course, and I'm not sure if you've read through the paper. I think it's the Ventura paper. I'll pull it up again. They go through a number of explanations as to why this.
We'll have to go through that. We'll have to go through that on some other point. Let me finish what I was trying to say before you interrupted me.
Go take your time.
I'll point out that Swamidass and Richard Buggs, as in examples of people that have written on this Eve issue, not one of them agrees with the idea that Eve could in fact have existed only 6,000 years old. So you don't have to worry about persuading me or any of the audience. You need to persuade them that your data point is right.
Well, wait a minute.
Wait for all this other material. You go ahead.
Well, RJ, we can move on.
Do your monograph.
But I want you to just answer this this question, because the thing is that this entire region in the chromosome 2 fusion, and you're the one who brought it up, that region that actually contains the reputed fusion site.
No, you brought it up this time, not me.
Genomes. But how come you're so resistant to just answer that question? I mean, we don't have to go.
I'll tell you. Jensen and company are wrong in their interpretation of chromosome 2. Lots of people with genetic degrees, neither of us have those, have analyzed this in great depth. I strongly recommend everybody go research this matter on their own. And you want to post the link to that paper, the Ventura paper. I don't have my bibliography up, so I presume I've got it in there buried somewhere. I may even have referred to it. Jackson and I may even have discussed it in The Rocks Were There. I'd have to go through my references on it. But I've made a big deal up front. I want to discuss your model. Nothing. Well, we say here.
Well, real quick. I don't want you slapping and running. RJ, we will get to it. This is our model, just like Kenneth Miller said. If there was no fusion, there was no ancestral dates.
I want you to apply it to a data feed.
I've been discussing this exact problem. Just a second, RJ. I know you're excited. This problem with the ape-specific satellite DNA that should be found flanking the reputed fusion site. I can screen share.
Who says that should be the case?
Okay, the ape-specific satellite DNA that's seen flanking chromosomes in the ape genomes should be, if there was hypothetically a fusion event after the split. How long ago, RJ, was the? Says, well, for example, this paper that I have, they're acknowledging that this isn't found and why, because this should be found.
And what explanation did they offer for it?
More or less the same explanation that I discussed it with Eric. I've discussed it with Ruhif. Ruhif says, he's got to relook into it. I don't know if the explanation they give is sufficient.
Well, wait a minute. You brought up the paper. You tell us, summarize what their explanation was for it.
Okay, so what we'll do right now is we'll give Standing for Truth a chance to summarize that. And so we'll maybe take like a minute or two. And then definitely, I understand if you have objections on each side. If you guys are able to give each side, your opponent, maybe a minute or two and let them finish, and then you can correct as many points.
I'll give him 5 minutes on it.
You got it.
Okay, what I'll do is, honestly, I'll just need 20 seconds to go over the argument. I'll go over how they explain it away, and then we'll see RJ's opinion on it, and then we'll go on to another topic. So, because we got so much to talk about, we only got 50.
Oh, yeah, including populating Americas.
Okay, so other than the area being highly degenerate, lack of cryptic centromere of the function, of course, chimps, most importantly, have very distinctive and very large satellite sequences, RJ, as you know, at the ends of their chromosomes, but they're absent in man, and these large ape-specific satellite sequences should be seen flanking the reputed fusion site if the human line and the chimp line split roughly.
Are you quoting the Ventura paper here?
Just to be sure that Standing for Truth had that point wrapped up, and then I promise we'll come right back to you, RJ. But you can correct as many points as you'd like at that point. I just want to be sure that Standing for Truth, were you finished with that?
Yeah, almost. I just want to make sure that, regarding this debate, I want to make sure that RJ has his own original thoughts, because I'm debating RJ and not the authors of these technical papers. So I just want to make sure that RJ has original thoughts and not just agrees with, his evolutionary side.
That goes both ways, Standing.
Now, of course. Now, the thing is, the ape-specific satellite DNA that should be found is completely absent in the region of the reputed fusion site. Now, let me ask you, when do you believe that this fusion took place? Obviously, we know it took place in the human line, but how many years roughly after the split between the chimp and human common ancestor? When did this take place?
If memory serves me, it's several million years ago.
Okay, how much?
Now, what does Ventura say?
So, it seems like there's a lot of mixed opinions as to when it happened.
Well, what does Ventura say? The paper you brought up.
Right. So the paper that I brought up here, I'm just going to pull it up right here. So the paper that I brought up, for example, the Ventura paper, let me go to it over here. They talk about how it is odd that this ape-specific satellite DNA is let me see, pull it up here. Real quick, have you read this ever? I don't expect you to know every single paper, but have you read it?
I'd have to check my bibliography. Ventura's in there. I wouldn't be surprised that it is in there because I've been keeping quite detailed track of this, but I can't recall instantaneously everything out of the tens of thousands of technical papers that I delve into.
Okay, and that's okay. I'll give you the title, and then we can just move on to another topic. It's The Evolution of African Great Ape Subtelomeric Heterochromatin and the Fusion of Human Chromosome 2. What they say is chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be composed primarily of arrays of tandem satellite sequence. And if you want to read all the details at the end in the discussion portion on how they interpret this, they'll say that due to the time, okay, and that's why I pointed out in my opening, the time dependency seems to be a primary rescue device for the evolutionists. Those sequences have been lost due to mutation and divergence since the split and since the chromosome 2 fusion. Is that an explanation you think that you would hold to? It's just been lost because it happened, you said what, 3 million
millions of years, wouldn't be at all surprised about that.
Right, and I would just ask, what testable predictions
do you have any reason to disagree with Ventura on that?
Well, I would say that it's just more so here's what I find funny, RJ. Here's what I find. If these ape-specific satellite sequences were found flanking the reputed fusion site, the evolutionists would be like, look at this, absolute proof for the fusion. Now those ape-specific satellite sequences are not found, so now they got to explain it away.
You're not claiming, are you, that that's the only reason to think that there's a fusion?
Of course not. That's why I said, hey, we could spend the entire debate talking about
right. And you will stipulate that neither one of us have written a technical paper published on this subject, and therefore we're both talking about it as non-experts, right?
Right. And I'll say one last point about this, and we'll move on to Eve of 6,000 years that you say nobody agrees with, including Swamidas. What I would say is, hypothetically, there could have been a human-specific chromosome 2 fusion according to our model, because we believe in a bottleneck at the flood. This would make sense given the data that there is no ape-specific satellite DNA found. I mean, that actually is a huge problem for ape-human evolution to begin with, and it's also a huge problem for the chromosome 2 fusion, but it doesn't disprove a human lineage-specific chromosome 2 fusion that's unrelated to ape. So therefore, if there was a fusion, it could better be explained according to our model. So it's 0 for 2 for the evolutionists. But like I said,
now I want to get to something else that's regarding your model. You're criticizing their model and offering a non-peer-reviewed argument, which you and Jensen and Tompkins all insist on. Can we discuss next, how did humans enter the New World?
Okay, so it seems like kind of a vague question. Like, for example, I'm using direct evidence. Geography is not.
What's vague? We got people
genetics.
Here.
Well, I'm saying,
when did they arrive according to
RJ,
your model?
I know you're excited. Just one sec. Mm-hmm. I gave a number of lines of evidence directly from because molecular clocks, for example, that can differentiate between the models. And guess what? The mitochondrial DNA, for example, and I showed the Parsons paper, the Gibbons paper, based on the actual empirically observed mutation rate.
Let's talk about that Gibbons paper. Is that the 1997 commentary in Science?
So would you want to skip past the Parsons paper and go to the Gibbons paper?
Well, it was a commentary on it. Are you trying to tell me that 1997 work precludes work done after that doesn't support that argument?
Every single, for the most part, pedigree-based mitochondrial DNA mutation rate study, including Gibbons, including the Parsons paper, I have a few more here. The FBI even adopted that mutation rate, takes us back 6,000 years to a mitochondrial leaf based on consistent mutation rates. How do you explain that data, RJ?
I may have to read the section I did on it with Jackson in the book. That may take a bit. Let me see if I can find that.
I mean, this paper right here, modeling the recent common ancestry of all living humans. The most recent common ancestor of all present-day humans lived just a few thousand years ago in these models. And I got all these papers. You've seen them in my opening presentation. Now, here we go. I'm going to ask you again, and I'll give you plenty of time to answer. Based on the empirical method, based on pedigree-based studies, okay? mitochondrial Eve goes back just 6,000 years ago. And Dr. Nathaniel Jensen said, okay, if this is true, the history of civilization.
No, it doesn't. That's incorrect.
I know you said it. I want to hear what you got to say. Just real quick.
I will not say anything other than it's wrong.
Yeah, but RJ, that's not an answer. So I want to give you as much time as you can.
Nobody in modern genetics, nobody, I know you like to repeat this.
Nobody will hear anything if
I'm peeved because this is annoying.
Okay, I hate
if
to
you're
do
going
this
to ask
you
me
guys.
like...
Hold on 1 second. Okay, forgive me for muting both of you, but just for a second, just to let's basically, I think that standing for truth, if you asked a question, unless it was rhetorical, like mention that it's rhetorical. Otherwise, let's let RJ answer the question, and then we'll come back to you standing for truth. Were you asking a rhetorical question, or was it?
No, it was a real question. Very simple, and I asked it in my opening too. Okay, how does RJ, we can talk about the Y chromosome after, but first we'll start with the mitochondrial DNA. How does RJ explain that based on the...
Are we going to go on this for the next half hour?
No. We're going to make it quick. I know what your explanation is, and then I'm going to ask you...
I've answered this question multiple times. All I'm going to say again is it's wrong. Mitochondrial DNA, mitochondrial Eve is not dated to 6,000 years by any current geneticist. Creationists claim that and use a variety of little circumlocutions. But I will not, I can't otherwise say that they believe something. Swamidass doesn't believe that. Richard Bugs doesn't believe it.
Have to do.
Why don't they believe it?
Because they know the data. It's their field.
No, because those that hold to the assumption in universal common ancestry, they're doing similar analysis.
Bugs doesn't.
Real quick, RJ, I'm giving you as much time as you need to respond, but all you're saying is they're wrong. If you want 3 minutes to explain the data, this is the observed mutation rate. People like Swamidass and people in these papers, right? The Parsons paper, the Gibbons paper, they're doing the analyses after the fact to calibrate it with the fossil record. But the thing is, here's what they're doing. They're using hypothetical mutation rates that are about 20-fold lower than what is observed.
So and as you know, insist.
Real quick, just wait, RJ. They justify this based upon certain evolutionary assumptions, and that assumption is that universal common ancestry is a fact. But the point in question is the geological column and the time frame. So all we need to use is the empirical method, and even the secular papers say Eve goes back 6,000 years ago. So how do you explain that without invoking phylogeny assumptions and so on and so forth? Take as much time as you need, RJ. Go.
I'm going to only say again, that's not the data point. You can insist that's what the facts are, up the yin-yang here. Nobody other than the creationists claim that mitochondrial Eve only dates back to 6,000 years. I can't say that. I will stipulate that I will say that a bunch of times, and you'll stipulate that you're going to say the opposite a bunch of times. Can we move beyond that now?
No, I want to screenshot. Okay, if you're going to make that claim, okay, and you're going to hold to that claim, I'm going to screenshot 4 papers that I have here. For example,
are we ever going to get to the New World?
We're talking about the New World right here because
we are. A good time to transition. So.
Yeah,
okay. I want to point out to the audience that so far on the ape-specific satellite DNA that should be seen flanking the reputed fusion site, RJ failed. He's also failed on the mitochondrial DNA. I've answered the New World because I said based on this observed mutation rate, if this is true and humans go back 6,000 years, let me ask you. No, I'm going to ask a question and give RJ as much time as he needs. RJ, you said you're up on the creationist literature. What testable predictions, okay, there's a few based on this, and it answers your New World question, is Dr. Nathaniel Jensen making? Go ahead, take your time.
He's arguing that Jensen claims that all of the DNA that he analyzes in various organisms, and it's not just humans, I got his whole book on the subject that I'm going through, supports a young Earth creationist model. That's his assertion. Nobody outside of that creationist field buys into that. And I know that Jensen is not a reliable source on these matters at the technical source level. That's an entirely different little area that takes a huge amount to get into. I want to talk about that New World that you have not discussed. Answer me,
I do want to discuss this directly.
Standing for truth. We've been on this a long time, so we are ready to transition.
I want to correct him because he gave the wrong prediction. For one, Dr. Jensen is saying,
James, covered this. Standing for truth.
No, I'm not going to move on to the next topic until you give me. I'm giving RJ as much time as he needs.
We just let you
go. Okay, well, I'm not moving on to the next subject until
wait a minute. You did not let him have as much time as he needed because you actually interrupted him while he was still talking. I would have believed you. I would have believed you. Hold on a second.
I interrupt him when he tries to move on to the next topic.
Okay, so even while I was talking there. Okay, so he was answering and you jumped in. So you didn't quite give him that much time. So we're going to go into the next one. If you would not have interrupted him, I would have believed you more, but you did interrupt him while he was answering.
Okay, maybe James, unfortunately you as the moderator, maybe you don't understand, maybe this is too technical. This directly answers his question. His prediction, Dr. Nathaniel Jensen, which obviously, unfortunately, RJ here didn't know.
We can go to the next topic, Standing for Truth.
This is the same topic.
I don't know if you're helping
Dr. Nathaniel
case.
Jensen is making predictions, genetic signatures in the mitochondrial DNA and in the Y chromosome regarding the new world, regarding the
that,
civilization. It's all there.
Can I get to one of my questions?
We'll go to the next question. Go ahead, RJ.
Okay, go ahead, RJ.
I am genuinely wanting to see Standing apply his creation model, which supposedly can make testable predictions and is terribly accurate, to that fundamental issue of who lived in the new world. Were there any human beings here prior to the flood? When did the flood happen? Guide us through the process of how Noah's kids extrapolated and then came to the Americas. What food crops did they have with them? What animals did they have with them? And justify that based upon the application of your flood model. Explain it. I am anxious to hear you.
Okay, so I mean that's like 5 or 6 questions already. Yeah, and I'll answer. So we now know that the mitochondrial DNA takes us back 6,000 years. So we know that history and civilization and people groups go back just 6,000. Okay, so now let's go in deep. Okay, and then we'll answer your question. Y chromosome now goes back only 4,500 years ago to Y chromosome Noah, which is also the topic of this debate. So now there's a 12-part series, and it take a lot to go over. Dr. Nathaniel Jensen's going over the people groups, the New World. Where have they migrated to, for example? Your question about were there people pre-flood? I mean, the answer is yes, of course. God created.
Were there any in America?
Well, I mean, America during the pre-flood world, during the, I guess, Pangea-like pre-flood supercontinent. I'm not exactly sure where Adam and Eve...
Put on your thinking cap.
Okay, so Adam and Eve...
You asked me, was I just going to be having my own thoughts as opposed to science works? Well, I asked you to do exactly the same thing. Don't trot out Jensen. You tell me what you think happened here. Tell us your model.
Sure. Okay, so I think that God created Adam and Eve with differences within themselves, and then this hypothesis, this created heterozygosity hypothesis, like you love, applies universally amongst species. We're talking about Adam and Eve. We're talking about Adam and Eve. So Adam and Eve were created, just 2 people, okay? Through these pre-existing DNA differences, they now have the capability of producing every shade of skin that we see today on the planet. Pre-flood world, supercontinent, as I would believe. They probably, I don't know exactly where, they lived. I don't think it would have been the Middle East.
Can you put a ballpark on it?
But we can look at Babel. We can look at the fact that Y chromosome variation is very limited, for example, and Y chromosome diversity around the planet is geographically specific. We got African Y chromosomes, Chinese Y chromosomes, American Y chromosomes. This is evidence for a dispersion and migration after Babel. I mean, we got the 3 mitochondrial DNA lines. We see this on every single continent. It's pretty easy to explain the people groups and the migration patterns based on at least Babel.
Really?
Pre-flood, I mean, I just explained
who's done that on the creationist side?
So, for example, using genetic signatures in the Y chromosome, Dr. Jensen's doing that right now. Have you read his newest paper on the Americas and the migration patterns regarding that?
Oh, make sure that's in their link there. I don't know that I've got that in there. What year is it?
It just came out, and he has one that came out in December, and he has another one that just came out. I may not be aware of that.
What is he saying about how many people were involved in the migration?
How many people were involved in the migration after Babel?
Into the New World, that specific group.
So in the New World after Babel, how many exactly? I'm not too sure, but all those DNA variants
was it 8 or 100,000 or what? What do you think?
Well, we know 8 people came off of the ark, so therefore,
okay. So it had to be more than that. Okay.
But if we started from 8 people, for example, and just 3 of those women, Shem, Ham, and Japheth and his wives,
Don't
they would have
forget
passed
Noah,
on their
Mrs. Noah.
mitochondrial DNA to everybody on the planet. And lo and behold, we see three.
How many people? When was this flood, by the way? Can you pin that down for us?
According to the Y chromosome and according to scripture, about 4,500 years ago.
4,500, not 2,350.
About 4,500 years ago.
Well, that's quite a wide margin. But let's go with the 4,500 years. Now, if we were to see the moment right at the flood and after Noah gets off, how long does it take before the kids of Noah, their descendant group, gets to the spot where they're coming to America? What kind of time frame are we talking about?
So, well, we would have from coming off the ark and going into Babel, we would have hundreds of years there, and then they would separate into, well, we would believe in the out of Middle East, and we know genetic diversity decreases every direction.
Talk about the Americas. Where are they fitting in?
We got
2,500, or we got 2,500 BC, and then that's the flood event. And so, when exactly are the Native Americans coming into the Americas? What time frame are we talking about?
Real quick. What relevance does this have to? Because I've already showed you that DNA variants, Y chromosome, mitochondrial DNA.
I'm going to get to the relevance. I'm trying to pin down this first.
Geographically, that's evidence. Now you want the specific details, and I'm showing you that somebody who's actually actively working in a research project. I've directed you to a 12-part series and 3 technical papers. Go read those after.
He apparently,
I don't know every single detail, RJ, of where.
Well, but again, I want to know what you think, not necessarily what Jensen thinks. You are capable of thinking on your own, right?
Well, I am, but I mean, according to your answers regarding mitochondrial DNA and the don't
jump back to that. Okay, now, have you ever tried to work out how many people could be generated from Noah and the kids over a certain time frame as a boundary layer?
I've read some technical papers on Robert Carter that looks at the HapMap data and the 1,000 Genomes project that looks at that. I've never personally done it myself, but I've read technical papers on it.
Okay, and so if I were to tell you that if you had an absolute upper margin of every single woman, including Mrs. Noah, giving birth to one child every year, regular as clockwork, and that 50% of them would be daughters, and that from the age of 15 on, each one of them give birth to people under the same circumstances, that in the course of 75 years, although you could theoretically generate close to 50,000 people, 99.9% of them are incestuous. Is that a workable dynamic for a population mix?
Well, it seems like according to population growth and how many people that we have today, that would only, and actually Dr. Jensen is proving this in his model, that it would only take about 5,000 years, and you could start from just a few people. But
has anybody fact-checked Jensen, including you?
Of course. I mean, all of his papers here on mitochondrial DNA that I've...
You fact-checked Jensen.
RJ, that's why I had about 10 to 15 papers.
No,
here.
You have repeated the claims he makes. That's not quite the same as fact checking. But anyways, I still want to get back to this Native American thing.
I want to finish your question about the current human population, because if you start from 6 people or 8 people, I guess, after the flood, and if you start doubling the population, let's say every 150 years, that more than accounts for current human population. That's a growth rate of what, like 0.5% year. And according to our model, there would be rapid exponential growth after creation and after the flood.
So you're claiming that the Native Americans came here like 300 years after the flood, or what?
Like I said, you can find that in his technical papers on genetic.
But you have not thought about this yourself.
No, I have. It's just if you want all those specific details, I'm showing you the bigger picture.
Did they? Why didn't they bring wheat with them?
Who says they didn't bring wheat with them?
There's no wheat known in the New World until the Columbians arrived.
Well, I would say probably just like the DNA variants that you see different mutations and different rare alleles in different population groups. Those are mutations after the flood. And when it comes to wheat and different food and sources like that, well, I mean, those are probably geographically specific as well. You're not going to find them everywhere on the.
You mean we can't grow wheat in Iowa?
Sure, we can.
Gosh, then why couldn't the Native Americans? And in contrast, how come potatoes aren't known in the old world until they're found from the new? Why the differentiation? Are you claiming that potatoes, rice, wheat, and maize, and chocolate and tomatoes and all that were aboard Noah's Ark, preserved, and so why the differentiation as to where they ended up?
Yeah, I'm not too sure exactly what food Noah brought onto the ark, but I mean, like I said, what's inherent?
But
I don't know how this is relevant to.
You have to deal with it as a problem, because by definition they had to have something like that if the model is correct. Why then the differentiation?
Why then the differentiation of food?
Yeah.
I mean, it depends. So obviously what Noah's bringing on the ark would be what was available. We know that the pre-flood world was green for the most part, 90% probably.
Including corn.
Vegetables. Well, corn, is that not a variation? Isn't that due to a specific mutation?
But it's also a food crop.
It would be like blue eyes, for
world.
example. Like blue eyes as a mutation.
There's no maize in the old world. It's all new world. Evolved in the new world.
Right. But the pre-flood world was absolutely destroyed. The real estate.
Yeah. So therefore, the only way to get corn would be to take it from a little box somewhere on Noah's Ark. How come only the Native Americans got that, but not somebody from China or Africa or the rest of Europe, where we know they can grow those crops?
Okay, if they brought corn with them, let's say onto the ark, would they need wheat?
Why not?
I don't know. I'm asking you, but I'm trying to show you a bigger picture.
I would have thought the most prudent thing would be everyone would take all the samples that they had, and that way there would be an actually global distribution of this, but that's not what we find.
Okay, see, the thing is, Dr. Jensen, as I said, he just discussed the Native American migration routes.
So he is your oracle here.
No, I'm saying that he has confirmed predictions based on the Y chromosome, which you're ignoring.
So you
we'll look at the details there. Now, wait. When it comes to Native Americans, they are actually Asians genetically that he is showing, and Asians didn't mess with, from what I know, wheat. They were more rice oriented. And you made one other point that I wanted to address as well. Writing systems, for example, Native Americans and Africans, from my understanding, they never really had a writing system. They didn't document their migration patterns, but like I said, you can see all the details in these papers, confirmed predictions based on the Y chromosome. I think I've answered your questions. When do I get to ask? Okay, when do I get to ask a question, James? I've asked.
I'm not through with mine.
I've answered 30 here.
I'm not through with mine. I want to know about the domesticated animals. Did they have horses with them?
Did they have horses on the ark?
Yeah, well, that too. What kind of horse was on the ark? And also, when did horse domestication occur in your model? And what was the Native American interaction with horses?
That's two questions. Okay, now a third question. I want to ask the moderator, James, because apparently I'm only allowed to ask 2 questions. You're allowed to ask 30. How many more questions do I have to ask, James, before I get to ask a question? Go ahead.
You started with 5.
So if it's been equal, we can go into taking turns, one from each person. If RJ's asked more than Standing for Truth's original 5,
I'll get back to him later.
Then we
can
go
do
back
that,
to
RJ. I have no problem,
Standing for Truth. Hold on,
I think I've answered all of them.
Okay, seriously, you just don't understand. When other people are talking, just wait for a second. Both of you this time. So basically, what we can do is, if RJ has asked significantly more than Standing for Truth, we can kick it back to Standing for Truth. But frankly, I haven't kept track of the actual number of questions.
Okay, so your last question had to do with horses. Okay, So today we know that there exists 850 breeds of horses and donkeys in the world today. This is your specialty. You probably know more than me. You've got the horse, donkey, and zebra family. I think there exists, 3 species of zebra, 3 wild asses, and 1 wild horse species, 7 species altogether. Now we know, and I think you'd even agree, that those 700 plus breeds of horses arose at some point during human history. That's obvious because humans bred them. Now there's only 7 species in the wild. So that's like 100-fold difference. You would say, according to your model, 4 million years for the wild, but apparently only thousands of years for the 700 plus breeds. Well, if Noah brought a couple horses, a couple archetypes onto the ark,
what do they look like?
Well, real quick, that had those pre-existing functional DNA differences. Those gene sites were heterozygous just through recombination, gene conversion, and other mechanisms of speciation, migration, isolation, inbreeding, of course, could easily lead to the 7 wild species we see today. I mean, we've got 850.
What about the ones that the Native Americans hunted?
The horses that the Native Americans hunted would have been some offshoot of the original, more heterozygous horses off the ark. Let me ask you, why are the 7 species of horses today, why are they more homozygous? Why do they have more homozygous gene sites than they do heterozygous gene sites?
Why shouldn't they?
Well, because that fits our model. We're trying to say, when we look back to creation, we're looking at the expansion.
Do you know what?
Well, you would be looking at the contraction. Well, real quick, you'd be looking at the contraction of the genome. So if God created a population of horses with these pre-existing functional DNA differences within and across, well, now, since it's been thousands of years, we should look at these species today genetically, and we should see that they're more homozygous. And that's exactly what we find. Shifts from heterozygosity to homozygosity.
Did the created horse look a lot like Dinohippus or something different?
Yeah, I know there's monobaramins, and I know Todd Wood is looking at that. It's tough when we get into fossils because, I mean, fossils, as you know.
What do you think?
Sperm and egg. Pardon?
What do you think?
Okay, so if we have 7 species of horses, and let's say Noah hypothetically brought 2, what exactly were the 2? I would say God obviously would have sent the most heterozygous probably on board the ark. Which one was it? I'm not 100% certain, like I said, because I'm only looking at fossils. So I can't give a definitive answer. I think Todd Wood has some good stuff on that, Kurt Wise, but it's not definitive because I agree. But fossils are tough. For example, how do you define a species just looking at fossils, RJ?
Oh, well, you can look at it in terms of morphology and that there are ranges on things that you can clearly move them into family categories. And then the genus area, there's very specific range precisely because the range between the extant horses involves other than just simple physical size, because you can breed very, very small horses and quite large ones. It's to do with the actual musculature and bone relationship. So it's a very technical discipline, which, by the way, the creationists tend not to deal with. I was trying to find your claim about the horses. Let's see.
Yeah, but real quick, because you said that the creationists are not dealing with it. Do you know how the creationists right now, for example, Nathaniel Jensen, Robert Carter, do you know how they're going about identifying the kind boundary and kinds in general? Because they're not looking at morphology and anatomy.
Oh, well, they've got a wacky arrangement that can never quite make their mind up on anything. They haven't been able to work out most of the kinds. They leave most of the data out. Todd Wood does that rampantly.
No, I know Todd
Wood. The data field down the.
Well, I know Todd Wood. And Kurt Wise, they look more to morphology, anatomy. That's why because it is difficult to give a clear definition of species when you're just looking at fossils. And the thing is, similarity in fossils may not always be due to common ancestry. That's why convergent evolution or loss of traits or incomplete lineage sorting is oftentimes involved. That's why we got to look at the DNA. And even Aaron Ra in his series there, he makes a point that dogs of all types make it clear that morphology and anatomy can oftentimes fail us. And why? Kind of like you said, there's often more diversity within a species than between a species. So that's going to make it difficult, RJ, to classify animals by their physical traits alone. That's why we got to look to genetics. My question to you with Nathaniel Jensen, Robert Carter, they're looking at molecular clocks and they're looking at DNA function to put a line in those nested patterns. Do you know the specific prediction that Dr. Jensen has made regarding the patterns found in the biological world?
The one that you have been repeating previously?
No, this is a different.
This is prediction. Tell us all about this prediction.
Okay, well, if you're looking at those nested hierarchical patterns and we're trying to say that those patterns are there for functional purposes, well, are you familiar with these highly, highly conserved mitochondrial proteins like the cytochrome C?
Yeah.
Okay, so why does, let's say, the CO1 gene, the cytochrome C, mitochondrial DNA protein, why, when you look at that genetically, why do they form nested hierarchical patterns? Why are there so many different
It's
variants?
impossible for them not to in a natural branching speciation.
Exactly. Doesn't that appear to be confirmation or evidence for descent with modification? Because all humans, how similar is the human and chimp cytochrome C mitochondrial DNA protein?
Off the top of my head, I don't know.
Well, it's very similar. And then the further you go, you can go to the old world monkeys, you can go down to dogs, cats, fish. Those
And
nested
your point
hierarchical
being,
patterns exist. So my question is, well, I guess if you don't know, Dr. Nathaniel Jensen has made a prediction as to why those differences exist, differentiating between nested patterns from common descent and nested patterns from function. So are you familiar with protein moonlighting, multifunctionality in these nuclear DNA proteins?
the term is vaguely familiar, but I don't know right off the bat on it.
I did show a paper. So mitochondrial proteins now have, it's kind of like a multifunctional tool. They're doing they're involved in not just obviously protein related function, but information flow and metabolic optimization, for example. So Dr. Nathaniel Jensen is making predictions that these highly conserved mitochondrial DNA proteins, such as the cytochrome C, that form these nested hierarchical patterns that seem to look like descent with modification. He's saying that those as well are multifunctional. Now, we don't have data to confirm this yet. Those tests haven't been done. So therefore, that is a novel prediction that can help us understand, is this descent with modification, or are these patterns reflective of function? And I can send you that paper too. It's about 50
I would love to. Just make sure that I don't already have it in the data field. Apparently, you're going to stick on the mitochondria a lot. Hopefully, we can get to mammoths and the Native Americans and those too.
No, I'm just showing you because you brought up the fact that creationists ignore the data field, and you always talk about Todd Wood and Kurt Wise. Todd Wood looks at morphology and anatomy, and Dr. Nathaniel Jensen disagrees with that because, like I said, well, I don't want to repeat myself, why bones are inconsistent. You can look to convergent evolution, for example.
Oh, but you will.
So they look to molecular clocks and DNA function. That's why the ALUs you talk about, or the ERVs, for example, the fact that they're highly functional DNA elements, that's certainly consistent with this model of, as you love to hear me say, created heterozygosity.
When
How do you
can we
explain that?
expect Adam
I'll ask
and Eve's
this one last
genome?
question, then you go back, and then it'll be your turn. How do you explain the functional roles found in these? I like to call them variation-inducing genetic elements. You'll say they're ancient remnants of viruses and parasites, but how do you explain the function?
What do you mean? How do I explain the function? Which one did you have in mind?
Okay, so if we're looking at, for example, why don't we look at endogenous retroviruses? Okay. So we know that they are they're playing important roles in determining.
You admit they're viruses, right?
Well, I would say I mean, like I said, I call them variation-inducing genetic elements based on their function.
Yeah, but are they viruses?
Well, I mean, we know that viruses, for the most part, within us are beneficial. We have more viruses than we do bacteria and cells. So, point is, are they created units of DNA as we would predict? And I would say that the evidence does suggest it. Now, you probably looked at the several papers that I had screen
Not
shared.
yet. I don't have the screen up to where I can see what those are.
Oh, you didn't see okay, so I had those screen shared.
Believe it or not, it's very difficult to just copy crap off that I see flip by at 900 miles an hour. Let me ask you this: you had all this information.
Answer the question.
Whoa! Let me put in. You already had this material prepared, right?
I had the sources prepared.
Okay.
Most of the questions
did you give James the listing of those references to post in the video description?
I will.
Good. James, hold him to it.
So. I could go on and on with these papers. I'll just read one line and then have you explain it. So this one says ERVs frequently act to distribute regulatory information and thus confer genes with new patterns of expression and function. So for example, would your argument be, okay, these ERVs that have crucial functions in regulating genes, determining cell types, they were co-opted? Like, do you have an original thought of your own, or is you just going to go with the author's conclusion?
I'll go with what the scientists say on it. It's their field. They're the ones with the PhDs. They're the ones with the actual pedigree. Am I supposed to override them? Are you going to override them because you have a dogma involved?
Okay.
As far as I can, let me ask you a question. As far as I know, all the endogenous retroviruses are called retroviruses because they start out as viruses, not as some originally created design thing. Now, if you want to present a monograph that establishes that the virus quality of them isn't in fact a virus, we look forward to your monograph on that. And don't just try to Jensen.
I appreciate you saying that. That's why I said the question is, are these really the remnants of ancient viral infections or ancient parasitic infections and invasions,
how
for
would you tell
example,
the
or are
difference?
they created units of DNA function? This is what I find funny, RJ. Okay, I'll ask you two questions, and if you want, you can go on to asking me questions. I appreciate that this is a little more respectful. Okay, so there's a class of retrotransposons. If you've seen my debates before, I've asked this before, maybe you're prepared for it. So this certain class of retrotransposons is found in the mouse embryo. Now, if you deactivate it, the mouse embryo will develop, and then suddenly it stops. Well, why? It's because it depends on the function of these retrotransposons, which evolutionists used to say were non-functional evolutionary leftovers, and supposedly these functions were co-opted. If that's the case, what, were mice just not having kids for millions and millions of years? How would you explain that? If these mice need these retrotransposons for development,
Who says
that's evidence of DNA.
It's the whole point is that lots of retroviruses, ALUs among others, can take on functionality. The question is, do all of them do that? I have a paper as well that I was just looking up today, infection by porcine endogenous retrovirus after islet xenotransplantation in SCID mice, where they were establishing that we need to worry about a lot of pigs as organ donors and others, because there are retroviruses in there that when transplanted into mice can take on their old viral functions and do bad shit. Well, it's perfectly understandable that such things
wait,
can
slow
happen
down.
in
Hey,
evolved
repeat that last part. What did
Was
they
that
do?
designed?
Well, wait a minute. What did they do? They implanted a virus into the mouse embryo?
They took the elements of the pig and transplanted it into without any modification, and
Okay.
it takes on its old viral function again.
What was the viral function? Something negative.
A lot of viruses, well, from the virus's point of view, it's all positive. But that
Well,
doesn't mean it's good for us.
But I mean, so could you, and you don't have to do it now, do you have any papers, and we don't even have to, it doesn't have to just be the ERVs, it could be any class of retrotransposon, any signs, lines, whatever, ALUs. Do you have any paper that actually shows empirically that a non-functional, whether it's an ERV, whatever, went from non-functional to functional? And then I can look at that and see, okay, here's.
Erica said you would bring that up, that
Yes,
was one of the tropes.
Okay, so what was the answer she told you to give me? Because if they're accomplishing crucial functions in regulating gene expression, differentiation, and development, I want to see a paper on how these functions can actually be co-opted, because it seems like the evolutionists, and unfortunately, RJ, it seems like you've lost one of your favorite pieces of evidence. So would you have a paper such as that?
That's the argument we go into, and this was not going to be a program on endogenous retroviruses, so I'm not prepared for that.
Okay, but
I don't have that at the top of my thing. This was about human evolution, Adam and Eve and Noah, and I've been hoping to get onto that world. The reason why I brought the subject up, in fact, was that nobody in the creationist worldview has thought about this matter of the population of the new world. And this new paper of Jensen, I'm going to be really wanting to see whether he does any better on that. They don't bother thinking through all of that, because the people who wrote the Bible didn't give a rat's ass for anybody out of their framework. All of that stuff didn't exist. They didn't even know it existed. So there was no framework for them to go on.
But here's what I find weird. You asked John, you've asked multiple creationists, and that's why I was excited to have this debate. You asked Adam and Eve, were they created with ERVs, ALUs, and these classes of retrotransposons? And now that I've cornered you on the topic, you're saying you're not prepared. Well, why did you ask?
I will go on at that issue about whether Adam and Eve had them. Do you know anybody, Jensen, Tompkins, Carter, Sanford, who has laid out what the original genomes of Adam and Eve needed to be in order to account for the heterozygosity of the hypothetical Noah and the kids, let alone all of the current ones? It's only 6,000 years. That's not that long ago. That's a relative snap for paleogenomics tinkering with that short of a time frame.
Yes, that's a good question.
I just want to let you guys know we'll go to Q&A in just a few minutes.
Oh, boy, we chewed up some time.
Well, has it been already 45 minutes?
It's going on.
How time flies when you're having fun.
Well, because I think, James, how many minutes did you give up, James, because we're having too much fun. So how many minutes did you give up to the
most of it. I think I only took 2 or 3 minutes to get my opening thing.
We're at about 43 minutes right now. So
can we go to...
We are close to that 45-minute mark. By the time we do a few more minutes, we'll be at about that 45 minutes.
Can we go to 50 minutes then, since James...
I'm okay with it.
I'm
Yeah,
okay
because
with
I'm
anything you guys say.
having fun. We get passionate, you know, and I actually like RJ. He's got good questions and he gets right into the literature like I do. But okay, so what was your question? Oh yeah, yeah. I was actually just talking to Jensen the other day about these variation-inducing genetic elements, the ALUs, the ERVs, and I was asking him, you know, how many of these do you think were created initially in Adam and Eve? But the fact is, and he directed me to a paper, a really highly technical paper that I can pass on to you. But the fact that these are all function.
And was that by a creationist or a regular science paper?
No, this one would be from Dr. Nathaniel Jensen. But the function is key. DNA function, molecular clocks. The function I've showed you that, well, I mean, for one, if viral DNA is inserted, as you were talking about, at random into the genome, and that's why I asked you if that change in the paper you were talking about was negative, it's more likely to disrupt existing genes. I told Conspiracy Cats this. I said if a blindfolded painter, he's going to most likely mess up the painting he is working on just by applying random brushstrokes to it. So these functional roles, there's no paper out there that shows them being co-opted. Therefore, we can safely say that all of these retrotransposons.
The reason why I bring up ALUs is that the vast majority of the ALUs that become functional are disease causers.
Okay, of course. I'm really glad you brought that up, actually. So, well, the ALUs are a little bit. That seems to be your expertise. So, you know, we'll stick on that. We'll stick on that there. So these ALU sequences, they're what? They're small repetitive elements representing how much percentage of our genome? 10%. 10%. Now they have the ability.
1.4 million of them and counting.
How much?
1.4 million of them and counting.
Okay. Now, because of from the papers I've read, and I've read a few over the last week, and it seems that they always assert. So they'll say in these papers, and once again, I'll pass those to James. They say because of their ability to move around the genome, these jumping genes are considered as real motors of evolution. That's all they say, considered as real motors of evolution when going over the functional roles that they have, because they talk about how the ALUs can allow cells to adapt to stress, for example, during viral infection. But like you said, they lead to disease. Well, that's what we would expect, James. If they're functional DNA elements and they're mutated, for example, or something negative happens to them, of course they're going to lead to disease. Would you expect it any other way?
Yeah, so when exactly would the first disease-bearing the reason why most ALUs don't do a damn thing is because they don't have a README code. That's why they're functionally invisible, and that's why they can replicate. That's why there's so many of them. That otherwise we'd be up shit creek. A few fitted into our brain chemistry. That's one of the notable areas where they took on a functionality. But most of the time, they're gumming things up because they're gumming up protein sequences. Now, is it a design defect of the designer to have put in a system that has a copy me signal like that just runs amok? Can we sue?
Well, I would say that copy me signal, those duplications assist in what I've been saying in variation. These are variation in inducing genetic elements. Is it reasonable?
Causing diseases.
Of course.
So the designer was okay,
RJ,
with causing diseases.
But DNA repair mechanisms, DNA redundancy, for example, requires forward thinking. And I asked Erica that question: how does natural selection, which sees the short term, not the long term, how does natural selection evolve something that requires forward thinking? How does natural selection evolve?
I think you may have an odd idea of what forward thinking applies in this case, and we're kind of far away. We use stipulate that no creationist has worked out what Adam and Eve's genome was. Is that correct?
No, actually, I'm going to send you. So Dr. John Sanford and Robert Carter are working on it. I think they've actually,
but they haven't done it yet. Is that correct?
Well, it's a work in progress. We understand very little of.
Is that going to be like Chadwick working out the paleo world? Is that a coming attraction that's never going to come?
I do want to answer that question, but I want you to answer this. Is it reasonable for us, RJ, to believe that these so-called ancient viruses and ancient parasites, the ALUs, possess the genetic information necessary to promote so many genetic functions and so many biochemical functions in human beings?
I don't accept your supposition. What about ALU do you think is like forward designing?
No. Well, you were saying with the mutations, for example, you're saying is this a DNA error, or is this an error in the design? Yeah, that's what you said. Sorry.
Mm-hmm. So you don't allow errors in design, right?
Well, obviously, if Adam and Eve were created with perfect genomes with pre-existing functional DNA differences,
right. You do not allow imperfection of design in principle. Is that correct?
well, no, I mean, we see defect. Like, for example, my car coming off the assembly line was near perfect. Now the air conditioner is broken down and rust is building up on the car. But
Which means,
that
as
doesn't
I
mean
said,
there was
that
no
you
designer.
do not conceptually allow for imperfect design in principle. I mean, it's a standard creationist.
No.
I don't know of any anti-evolutionist that.
Yeah,
that
but
would
you're
disagree.
familiar with genetic entropy, RJ. We don't have to go down that route. I've said this before, so you're kind of, this is going to be a rabbit trail, but we accumulate 100 new mutations per person per generation. Take that accumulating mutational load back to a point of least mutation accumulation load. That's a point of perfect genetics, Adam and Eve. So yeah, after the fall, mutations have been introduced into the genetics of people and species. And now these ALUs, these ERVs, if they're subject to mutations, that will result in, of course, disease. But before we end, you asked me, have they worked out the original genomes?
Yeah.
Okay. Well, that's why I went over I didn't have time with you, but I've asked before. They're looking at, because we know human chromosomes are made up of relatively large linkage blocks. And Dr. Sanford, and you can look at this in the HapMap data, they're looking and analyzing these original text strings. And it shows us that the genome is young. Why? And then I'll let you respond. Because the genome has only been partially scrambled. When in fact, RJ, crossovers and gene conversions should scramble all linkage blocks quite quickly in an evolutionary time frame. That's a huge problem for you. Answer that question, and I'll tell you how they're using that data to come up with the original.
Who says that that's the problem? Is it stuff that Sanford says it's the problem, or do you have a regular scientist that says it's a problem?
Good question. Regular scientists, and they have explanations for it. Do you know what the explanations are for it?
Oh, well, summarize it, because right off the bat, I guess so. You summarize in the way that you didn't summarize Ventura's paper. Summarize this one for real.
Well, I did summarize the Ventura paper. They say that the sequences have been lost due to deep time dependency did it.
Oh, okay.
As the evolutionists would say, God did it. Okay, well, real quick before we end. So they're looking at those linkage blocks, and since they're still so large, that means there hasn't been much gene conversion or recombination going on. They can actually determine the original chromosomes, the original linkage blocks, and they're doing phenomenal work in it. And Dr. Jensen's looking at it through mitochondrial DNA differences, and they're close.
We'll see.
They're close.
Do we still have enough time to talk about mammoths and the New World?
We got about 30 seconds. I mean, I can go longer if you guys want to go a little longer, but just so you guys know that, I mean, like, we're at that originally planned time. But yeah. I'm flexible if you guys. I mean, we could.
Well, I know RJ likes a long Q&A though.
How about if we go to Q&A, get that out of the way, proceed on that, and if Standing and I are still up for going on, we can resume a conversation as long as you want to, James.
I want to say one thing. I like talking to RJ. I would say he's one of the more formidable opponents, if you want to say that. He knows his stuff. So therefore, I would even have additional discussions on his channel or my channel, since there's so much to talk about. Right now, maybe we should go to the quick closings and then a long Q&A.
We want to hear James have to pronounce some a ridiculous name and be embarrassed by what he has to read in the super chats.
So true. And we have plenty of trolls here tonight. Michael Dresden, our regular, he's a character, says standing, I think he said this before the debate started, maybe. Said standing for truth already won. Case closed, people.
I think that's a repeat of his last super chat.
He's
James, I think he's got it on cut and paste.
Yeah, I think you're right. Wikey9, thanks for your super chat, said amphibious vehicles standing for truth just went full crocoduck.
Right. And, I was thinking if RJ had a problem with my explanation for transitional forms, he better call the military and tell them to stop building amphibious assault vehicles, because they're making it too easy for us design advocates.
You get an amphibious vehicle that replicates and come back to us on that.
Gotcha.
Hey, adding reproduction only makes it tougher for your
kids. You don't want to see amphibious vehicles mate.
Thank you. That's quite the picture, RJ. Thank you. Next up,
You're
Vodka
welcome.
Vision. Thanks for your super chat. Said, why can the theory of evolution predict both extinct and extant species before their discovery? When will creationism be able to do the same? Standing for truth.
You know what? Unfortunately, I'm going to have you repeat that, because it's something you cut out there.
No problem. Vodka
we're
Vision said,
distracted.
why can the theory of evolution predict both extinct and existing species before their discovery? When will creationism be able to do the same?
Good question. Good question. So evolutionists have had so many failed predictions. They've made a few lucky guesses based on the fact that, hey, listen, if you stick me next to a midget and then a tall 8-foot basketball player, you're gonna have a few lucky guesses that, hey, there's gonna be an intermediate found, or a tricycle, a bicycle, and a four-wheeler. Sure, there's gonna be some tricycles out there somewhere. You can find them in the junkyard too, probably. But we're actually taking these predictions in genetics. Genetics is what's inherited sperm and egg, and we're making the predictions on speciation rates, mutation rates, DNA function, but very specific predictions on DNA function that I'm actually shocked that James wasn't able to reiterate, because he says he's up to date on the creationist literature, which means I'm excited to send him all the papers I have.
Gotcha.
On that point, I'll point out that geneticists, I can't remember there was a bird species or a lizard species. I think we mentioned it in the rocks were there. But at any rate, that it was clearly a hybrid form, and they had the genome of the putative species that was the one parent. They were able to genetically work out what the other hybrid needed to be. And it turns out later on they found that that exact species actually existed, and so they were able to identify the parentage lineage. There's an awful lot of stuff that goes on in this field, but it doesn't show up in the creationist literature, and therefore it doesn't get filtered through into the Jensen stuff that Standing is so good at replicating.
Wait, I just because this super chat was originally meant for Standing for Truth, I got to give Standing for Truth the last word just so we don't gang up on him. If you want to say a quick rebuttal to RJ, you can. Standing for Truth. Then we got to
no, because to be honest with you, I was a little distracted here. Like I said, my wife's getting ready for work, so I didn't hear much of what he said. So
families take priority, Buckaroo.
Go ahead, next question.
Next up, Terry James, thanks for your super chat, says creationism does not predict nested hierarchies. If nested hierarchies didn't exist, that would be used against evolution and for intelligent design.
Right. So. For one, Linnaeus, he's the father of taxonomy, kingdom, phylum, class, order, family, genus, species, and he based his classification system on function. And we're predicting that these patterns that are found, they are there for functional reasons. And I gave very specific predictions on those highly conserved mitochondrial DNA proteins. Like people like conspiracy cats or Herman Mays will say, why did God create so many variations of, that same highly conserved mitochondrial protein that does one function? And it's like, no, these, we're predicting these are multifunctional. But of course, we're going to predict more similarities in anatomy, morphology, and genetics and physiology with a human and a chimp than a human and a fish. And we see the echo in the design realm with modes of transportation. So expectations on both sides, but here's the key. Predictions from our side, no predictions from the evolutionist side.
Well, that's a claim that you're making, but I read the evolutionists and the field in there. And the point about nested hierarchies is that there is an inevitable discontinuity between the alleged kinds. And the problem is that whenever they try to look in the details, they have to start mugging up the boundary layers to avoid creating too many interconnected kinds. That was the problem that Wood got into by including all the way back to Heracitherium on the horse sequence. And that very mechanism, if applied to the human holobaramin, puts up Australopithecus sediba in the human holobaramin, which Wise got a bunch of pushback on that one. Wise or Wood, whoever had done that. I think it was Wood that did the study on that. So they're way behind the actual systematics in this area, and they've been that way for an awful long time, and I don't expect them ever to catch up.
Gotcha.
Was that super chat for me, James? Or?
It was. That's why I'll give you a quick response, and then I've got to try to ask you guys as hard as you, if you can exercise your self-restraint as much as possible. I know you've already.
That'll require a miracle.
You've got
James.
two talkers here.
You've always got another round in the chamber ready to fire, but just to get through as many questions as possible. We'll give you one rebuttal, Standing for Truth.
Yeah, real quick, 20 seconds, not even. I think James proved simply by me asking a question, how do you define species using fossils? And it's very difficult. And of course, similarity doesn't always mean ancestry. That's why they invoke convergent evolution. I can go on and on and on. But we, using DNA function and molecular clocks, for one, we've already separated humans. I mean, humans are not related to anything else based on mitochondrial DNA function, and Y chromosome. And we're doing that same thing and applying it to all the other species. Maybe, RJ, our next debate can be on species and not humans.
Gotcha. Thanks for your super chat from our dearest friend, Terry James. Oh, we got that. Sorry. Dwayne Burke, thanks for your super chat. Says, RJ, all humans are traced back to mitochondrial Eve, but Eve had a mom. So why don't we stem from Eve's mom?
Well, that's Swamidas could probably explain all the little ins and outs of how you identify that kind of a boundary layer, that you can push it back. It doesn't necessarily drop off at exactly, within one generation of it. It's just a ballpark figure that you can deal with. And the point is that you can generate both male X and Y chromosome lineages that will overlap and involve more than one progenitor, depending upon which one you're looking at. The point is that none of them actually match up with that 6,000 time frame, or for that matter, with that 4,500 time frame, although obviously Jensen and company insist that it does, and Standing agrees with them.
Gotcha. Thanks for your super chat. If you want, since I let RJ do it, I'll let you do it, Standing for Truth, as long as it's short and pithy.
I appreciate it, James. I do want to say you did a good job moderating today. I'm sorry I got a little heated there for a second, but what can you expect? It's me and RJ.
Yeah, okay. I know. We both of us have a thermostat that gets up.
Not a problem.
Especially when I had a coffee.
So your guys' patient, or your passion.
Oh, yeah.
Go ahead.
Real quick, I just want to say, according to the evolution model, there was really, at the end of the day, every possibility that we would have found multiple diverse mitochondrial lineages within modern people. And some of those, or even all of those, could have been shared with chimpanzees, but yet it just fits exactly what we would expect based on the account in Genesis. So I think it's fascinating. Thanks, James.
Gotcha. Thanks for yours. Oh, that's right. RJ, that was originally for you, so I'll give you a chance to do a rebuttal to Standing before you go to the next one.
Given that millions of years involved, none of the features that we see and the amount of degradation that can occur in a natural lineage over time, and the generation of new elements over time, are ones that cause the people working in the genetics field to have conniption fits. The main thing is they're applying the work. So they work out population distributions and haplotypes, and they're able to work back in various regions to find out what intermixtures have gone on over various times. And when the creationists get to the level where they can actually persuade people outside of their box that they're right, then pop the champagne corks, but not until then.
Gotcha. And thanks for your super chat, Robert Lescombe, who says, I can't read Spanish that well. James, I have one question. I only want you to speak Spanish. I can only read a little bit in Spanish. No tengo una my Spanish isn't good. No tengo una pregunta. I have no question. Solo quiero. I only want escucharte. I don't know that word. That one's new to me. Hablar español. So speak Spanish. Thanks for that, Robert. Very let me know what escucharte means and how to pronounce it. Next up,
Terry throws in Urdu. Run for your life, James.
Huh? Is it something sexual in nature? Is that what you said?
No,
RJ?
no. I think it's a language in Iraq or something. It's a Middle Eastern language.
Gotcha. Thanks for your super chat. Holy skepticism is coming at you today, RJ. They said there's zero, in all caps, evidence for evolution, period, zero.
Yeah, well, I'm sorry, but I see a lot of it, so I'm afraid I can't flush it down the toilet just to make you happy. But if it makes you happy to think there's zero, then go ahead.
Thanks for your super chat. Terry James, who said, why didn't the designer make animals that defy nested hierarchies like a griffin or Pegasus?
Well, I mean, that's why I discuss things such as convergent evolution that they have to look to, for example, or loss of traits, evolution. For example, look at the living duck-billed platypus. That creature is a mammal, and yet it has a duck bill. It's got webbed feet, lays eggs.
Not quite.
And real quick, I knew this one would excite you, RJ. I'll give you the last word on it. But in addition to having those characteristics, one might even call them reptilian. and it has the characteristics of mammals, reptiles, birds. I mean, give me the family tree of, the single-celled ancestor billions of years ago up to the platypus, RJ, go.
Yeah, the bill of the platypus is not like a bird's bill. It's structured completely differently. It's not a hard keratin thing. It's not a bone structure. It's a fleshy flap. It has electroreceptors. There's a huge amount on its evolution. I discussed quite a bit of that in Evolution Slam Dunk. I think we brought it up along the way in The Rocks Were There. Book plug. And the monotremes being the egg-laying group, things about the immune system, the developmental placental systems out of egg-laying monotremes, all of the complex biology that leads to the marsupials and then on into the placentals like us. They're an absolutely fascinating area of study, none of which has been done by anti-evolutionists.
Gotcha. And I'm trying to remember who that was originally for. Standing for truth, if you want to give a pithy rebuttal, I'll let you.
It's tough to give a rebuttal to that because that could probably take an entire debate. I mean, it seems like it'd be even tough for him to give me the absolute evolutionary process, because it seems like the platypus appeared very recently, like maybe 150 million years too late to even be the ancestor of mammals. And we could probably go on and on and on about.
Yeah, we know.
See what I mean? So we could have a whole debate on this one.
I'd have to look through my things on the fossil
I find
record on the
platypus
monotremes.
interesting, though.
Gotcha.
Go ahead.
Let's see.
Yeah.
Thanks for your super chat. Sorry, I was distracted for a second. And Zekus, thanks for your super chat, said, standing for truth, the 4th dynasty of Egypt, the zenith of the Old Kingdom, was smack in the middle of your flood. All of the famous pyramids were built within plus or minus 100 years of this time. The flood didn't happen.
Yeah, I mean, there's been tons of articles and papers written on this. I'm surprised they even used that same argument. We know that according to the Egyptian king list, RJ, you're not going to deny this. I mean, the pharaohs.
Oh, yes, I am.
But the pharaohs themselves were oftentimes dated according to the pharaohs overlapped themselves in reign, but then they didn't consider that, which means that their dates, their calendars are way too off, much longer than it actually is. But I mean, you can look at the languages and written records and all these things. I mean, just Google it. Even the secularists will say it only goes back 5,000 years. So just suddenly language exploded on this planet. No. All the Chinese civilization, all these different kingdoms, the Egyptians, they go back just to after the flood. The timeline works perfectly.
Gotcha.
And I'll say that this has been a ticking time bomb. I'm older than you are, Standing. I can remember all those decades and decades of creationists not thinking about the damn Egyptians because they aren't Egyptologists and they didn't pay the slightest attention to when the pyramids were made. And I was just waiting for some of them to realize, the Egyptians are straddling the flood layer, and there's no way they're going to do it without moving all of Egypt down. So what you're interplaying here, Downs, there's a few small others. Elizabeth Mitchell riffs off of them the same way. They are trying to scrunch all of Egyptian history down, including the pre-dynastic stuff after the flood. And it is an utter mess. We're going to bring that up in volume 2 of The Rocks Were There, because it's all part of that flood chapter thing that was going to be chapter 8 of the book, but now is part of the next volume.
I'll give you a quick response. Standing.
Thank you. Yeah, no, I've got some pretty extensive articles regarding the history of man, all these civilizations. It looked like RJ didn't deny the fact that these pharaohs were recorded in a linear fashion when in fact they overlapped, which means those dates are
not the Old Kingdom.
Well, I don't know if you've seen the latest 2 documentaries, Exodus, for example, that actually lays out the dates that seem to fit in accord with Noah's flood. But yeah.
Oh, and who did that one?
Next, we might have to keep
evolutionists.
Thanks for your super chat from Wendy Lynn. Appreciate that support. Didn't see a question, but if you had one you wanted to attach, let me know in the live chat. Thanks for your super chat from Terry James. Says a supernatural designer can create things from scratch, unlike human designers.
I'm guessing he's saying should, because we see similarities, homology, nested patterns. Like I said, if he's create from scratch, I mean, God did create from scratch, but he used similar features. He used a similar genetic code, mitochondrial DNA code, the nuclear DNA code. But I mean, I've said it many times that based on the design model, we as creationists, we want to get a sense, okay, how did God create life? Therefore, we look to human designs. And it just so happens we don't even do it on purpose. We don't even try, but it turns out that we design in those nested hierarchical patterns. So if the evolutionists have a problem with that, if RJ has a problem with that, then he should have prayed that we didn't design in that type of fashion that just has an echo in the biological world. So
gotcha. Thanks for your super chat from Drew Rayford. Has my back. Says, standing for truth, don't insult the moderator. It's rude. Thanks for that. I appreciate that.
He controlled the mute.
It's to be fair, I appreciate Standing for Truth's sincere apology. I don't mind the passion. I appreciate the passion, and we're happy to have our guests here. That's why I always try to, like, I know sometimes it's like, James, step in more. But it's kind of like, well,
and I can tell you, you have the right to do that, James. And so, you can have it's okay to have gumption on there and say, bag it.
I will, yes. I will. I'm doing a little more actively than, like, let's say a month ago, but yeah. I'm still trying to be pretty deferential because I always just want the debaters. I feel like the more freedom you have, the more fun you feel, and the more fun the debaters have, the more fun everybody has. So,
but both of us can run off at the mouth, so you got to bear that in mind, James.
I feel like RJ and I need about maybe a 6-hour discussion portion. But James, this is your house. This is your channel. You're doing a great job. Congratulations on the 20,000 subscribers. So yeah, like you said, we get passionate, and anytime I apologize, I appreciate the. You're doing a good job, brother.
No worries. And we are excited. I do want to say, if you hadn't seen me, I got on I think it's like the Friday night stream the other night just to say thank you so much, folks. It's exciting to hit 20,000 subscribers. It's a fun community. We really hope it is eclectic. We hope it's a variety of people from walks of life. And so we really do. It's always fun. You guys make it fun.
Just watch out if you ever hit 666K.
That's true. I will. I appreciate it. Always looking out for me, RJ. And
yeah, I always am.
Kevin Guilfoyle, let me know if I know I mispronounced that. Just let me know how I can say it.
Guilfoyle, maybe?
Thank you for your super chat. Said, standing for truth, why do you cherry pick data from non-peer-reviewed sources and then misrepresent them? The real data proves you wrong. RJ totally won this one.
RJ hired him to say that. No, I'm just kidding.
I'm sure Standing will disagree and demur.
But do I really need to screen share? I mean, I had to choose between hundreds of papers just even put into my opening. If RJ goes back and rewatches this debate, he's going to see. I mean, these are all.
I just asked that you make a point of putting the sources up in the link in the video chat. So anybody, because it's not just me, anybody watching the video should be able to look that stuff up.
Well, I'm going to put a challenge out there to anybody. If anybody wants to debate mitochondrial Eve, and we look at these papers and the observed empirical mutation rate, these pedigree-based studies, they all go back to an Eve just 6,000 years ago. And I will debate anybody on that because it's on our side and we're the ones making future predictions. On Dr. Nathaniel, I say we, but Dr. Nathaniel Jensen made them on the Khoisan peoples. Hey, pointed at these people.
Erica said you'd bring them up too.
Gosh.
RJ, you should have had an answer prepared if she warned you.
Because I wanted to discuss the Native Americans, and I learned what I wanted to on that.
We did, and I'm going to send you those papers too that goes over it in more detail. Sorry, James, go ahead, brother.
No problem. Next one, I've got to wrap up for sure by 9 p.m. my time tonight.
Oh yeah, before 9 because there's an after show afoot, so that's at 9.
Ooh,
thanks for your super chat.
But that's another hour from now.
You got it. And Coco Puffer, thanks for your super chat. Said, "Hey RJ." Quote, in all caps, seeds can float. Duh, Noah didn't need to bring all the crops in existence with him.
Well, let the creationists know that. They seem to have a slightly different impression of it.
Gotcha. And thanks for your super chat. Michael Dresden. Oh no, here he is. He's back. You heard from him earlier. He says, man, this is sad. RJ lost and doesn't even realize it. Another one of his standard copy and paste. Go ahead, RJ. If you want to respond, you don't have to.
Nothing need be said on that.
Gotcha. Thanks for your super chat.
Actually, James, real quick, and take as long as you want. We didn't do our 3-minute closings, and I'm not saying I need 3 minutes. when the Q&A is done, can you just give us even just anywhere between 1 minute and 3 minutes just to wrap up the conversation? I just want to explain how.
Yeah, I'm okay with that.
That's
yeah,
true.
We can zip it up then.
We've got okay, so why don't we do this?
A closure.
We've got a number of super chats left. Let's do those closing statements, and then we'll come back to the super chats and fly through them as quick as possible. Because I did promise those closing, so I do want to keep that promise. So let me set the timer, and if I remember right, RJ went first. So RJ, I've got the timer set, and the floor is all yours for your closing. I'll start it on your first word, RJ.
Oh, the basic thing, the reason why I wanted to bring up the human evolution in the human context in the North American and American context is because I know what the creationist literature is on this, and they've avoided the subject. And I was curious to see just how much Standing could wing it to think on his own, to work out what his own model is. And it's incredibly vague. That whole sequence of how organisms developed and how the population spread out. He's got the mantras of the mitochondrial DNA and the heterozygosity down, but he can't apply that to how many did what, with what, when. We didn't even get to the mammoths because we would have to figure out why there were mammoths in the New World and are they post-flood or not. That's a whole other can of worms, but we didn't get to that. So I learned what I wanted to know in my end of the discussion.
Gotcha. Thank you very much. And Standing for Truth, the floor is yours for your closing.
Awesome, thanks. Yeah, I'm not going to take too long here. I'm just going to share screen again real quick on the, not sure if it's working, just on some of these papers that I have. Yeah, I mean, these are all secular sources, for example. I mean, even the Parsons paper, the mutation rate derived from there, the FBI has adopted it, but according to the evolution, it's still not good enough. And they can say that the mutation rates were slower in the past, for example. But the question is, when does the molecular clock speed up and slow down? I asked that question 5 or 6 times to Erica. We discussed it for 30 minutes. She admitted there are no predictions, but the creation scientists are making very specific predictions on the rate at which people groups, for example, the Khoisan peoples over in Africa, the rate at which they mutate, they change per generation. So, and he's made predictions on the history of civilization, and that's what I was saying. He wants to know all the details. Well, we know molecular clocks take us back just 6,000 years, 4,500 years to Noah, Y chromosome Noah. So we know that all of human history fits within that time frame. Now we're digging deep. We're making predictions on the history of civilization. And Dr. Jensen has a few papers regarding the history of civilization. I believe I did answer all those questions that RJ threw at me. I don't believe he answered really any of the questions regarding the molecular clocks, chromosome 2 fusion, and the DNA function. DNA function is huge. It's huge. And, his position was human evolution. He brought up the Australopithecines. I think I refuted that. And just in general, I'll end it with the fact that any of these so-called pre-human ancestors that evolutionists will point to as our so-called answers, for example, Homo floresiensis, right, the Hobbit, Erectus naledi, Heidelbergensis, Neanderthal, they were not actually pre-human, and that even those so-called pre-humans are better understood, according to our model, as human people groups who underwent accelerated genetic degeneration due to severe and extended inbreeding. This is all because of post-Babel migration and isolation. I kind of wanted to discuss that with RJ, but overall, I really enjoyed the debate. Overall, I really enjoyed the debate, and I think it's pretty evident that the creation model came out as the stronger model today. But thanks so much, James, and thanks so much for James too. That's it for me.
And thanks again to the speakers, who I have to mention it because I just appreciate these guys, and they're always fun to have on. They're passionate. Their links are in the description, so if you're listening and you're like, oh, I desperately want to hear more, you can hear more by clicking on those links that I put below. So we'll jump into the next question. Appreciate P. Barnes, thanks for your super chat, who said, saying for truth, why do we need to do gene pool mixing to conserve animal populations if humans turned no genetic diversity into 7 billion people?
I think I understand the question. I don't know if he's saying that there's too much genetic diversity in the world population today to come from just two people. But I mean, I answered that objection in my opening. That assumes that all DNA differences, all diversity is the result of mutation, but we invoke created heterozygosity. I'd like to make RJ a shirt that says created on one side, heterozygosity on the other. I know he loves that term, but yeah, that makes predictions that flow from it. It's not just ad hoc, post hoc. We're looking at DNA functions, speciation rates, mutation rates. We touched on a number of those predictions today, and I'm going to send those papers to RJ. And as he said earlier, he wasn't prepared for that. Maybe next debate he will be, and we'll have another solid debate. So.
Gotcha. Thanks for your super chat from Josh, who says, James looks like a supervillain tonight. The light coming from below makes his glasses cast crazy, in all caps, eyebrows on the forehead. I heard that. I heard they look like supervillain eyebrows, like you said, Josh. Thank you for that.
All you need to do is to practice a Boris Karloff impression, and you can have Halloween covered any year.
Everyone keeps saying that. Who's Boris?
Boris Karloff.
Is this like one of your personal friends, RJ?
All you got to do is, I've no doubt about that, but if you need, the impressionist Rich Little said, if you ever need to do a Boris Karloff impression, just say, antipasto.
Gotcha. Next up, thanks for your question, Adam Elbilia, appreciate it, said, calling convos like these, quote, debates, unquote, is nonsensical. You can ignore the facts, but sciences and pseudoscience is not even on the same level. Nonetheless, these kind, in all caps, haha, get it, of conversations are usually entertaining and enriching on both sides. So, hope you guys appreciate that compliment there. Puts a
Very
little
sweet
spring in your step. And thanks for your super chat as well from Kevin, who said, standing for truth, why is it so hard to say, quote, I don't know? You obviously didn't know the answers and instead made up BS. Just be intellectually honest. RJ won.
If you're referring to the more questions he had regarding the negatives and the migration patterns, I think I answered them quite sufficiently. And I think according to the bigger picture regarding molecular clocks, for example, that points us to a history of civilization that only goes back 6,000 years. So yeah, so some of those details I'm not going to know right off the bat, but like I said, there's a 12-part, I think it's up to series where Dr. Jensen, who the evolutionists hate, but they can't refute him, goes over all these genetic stamps that he's found in the Y chromosome. If evolution was true, deep time evolution, those genetic signatures should not be found because 250,000 years worth of human evolution, for example, it should just be a big scrambled mess. But yet we're detecting signatures, proof. Science doesn't use proof, but it's a testable, confirmed prediction based off our model. So,
gotcha.
I'll just say that when Jensen manages to convince somebody outside of the creationist community that his interpretations are correct, send us the memo on that.
Quick one, standing for truth, then we must move on to the next one.
No,
We've
that's
got
okay.
1 minute left. Okay. Pete, thanks for your super chat. Said, standing for truth, what research did you perform after your debate with Erica? Seems like you're on the same script.
Oh, you mean that debate where I asked her 5 or 6 times in the first 30 minutes? And I just want to say I really enjoyed my debates with Erica. But yeah, you know what? No testable predictions, non-science. So I asked, when does the molecular clock speed up and slow down? She's looking to slower mutation rates in the past, but she can't make any testable predictions. We look at the empirical method. The observed mutation rate takes us back 6,000 years to Eve. Evolutionists now have to calibrate the data with their assumed deep time evolutionary history. It's not science unless they make a prediction, which they won't. Erica admitted she didn't. So
thanks for your super chat. So sorry. I can't let you guys do any more rebuttals because I have to get out of here in about the next couple of minutes or I'll be in trouble.
No problem.
Thanks
Or
for
you'll
your
burst into flame.
We're always happy to have you guys back, so keep that in mind. Thanks for your super chat from Dwayne Burke, who said, RJ and Steve McRae are brothers, just saying.
Oh, I hope not.
Gotcha.
I thought they looked a bit alike.
There's something very strange going on in our family of so.
Coco Puffer.
No offense, Steve.
Coco Puffer, thanks for your super chat. Said, hey, RJ, Dr. Jensen has a PhD. Yep.
Oh, yeah. So does Tompkins. So Sanford, they've all got PhDs. That's no guarantee that they're correct.
Thanks for your super chat, Mothra J Disco as well, who says, Kennewick man legit debunks Standing's time frame. Did I
Say
pronounce
that again, Jay?
Did I pronounce this right? They
Kennewick
said
Man, yeah.
Kennewick Man legit debunks standings time frame.
Who, man? Oh,
Kennewick Man is a ancient
yes. No,
Native
I know.
American that was
Yeah,
found in my
Raw
neck of the woods
Right.
in Washington State, and dates back many thousands of years, and was an interesting cause celeb over Native burial rights and a whole bunch of stuff. And that's all part of all of that information that I'll be anxious to see whether Jensen ever accounts for it.
Well, you can even look in Raw Matt and myself and some of the things that we've got on our channel. We have addressed that specific one, and it still lines up perfectly with the 6,000 year time frame. So
Everything is going to line up perfectly if you pair off enough data.
Gotcha.
near perfect.
And thanks for your super chat from Speed of Sound of Gravity. They said, this is the last of the money from my son's new shoes fund. Giving it to you, James, for telling Standing for Truth to shut up. Gotcha. Thanks for that. Very sad.
But the little kid's going to go shoeless now.
Yeah, but James, you tell me to shut up every debate. This is nothing new.
That's true.
That's true.
So, yeah,
And if I don't get asked to shut up once in a while, then I'm not doing my job.
It's my mission. James has to get me. James has to tell me to shut up once per debate. That's my mission.
It's a tradition.
Got a little quota.
Fat Man, thanks for your super chat, who says, Dwayne Burke, if they are really brothers, it makes sense because they're both very smart. Oh, well, nice compliment to you and Steve McRae.
Oh, shucks.
Thanks for your super chat. Lily R.O. who says, James, do you guys have merch? I'd buy a calendar of you. Oh, gosh. Okay. Lily R.O. is secretly Tom the plumber from Texas. So it's appreciate that, Lily. Thanks for your super chat. General Ballsack has struck again. He says, none of you know how amphibious vehicles work. Thanks for your super chat from everybody's just shocked.
Assault vehicles.
Thanks for your super chat, Disco Plumber, who says, James, is that your kill room that you're in? Scientists are saying the future will far more futuristic than they originally predicted. I think they mean be far more future. Scientists are saying
future isn't what it used to be, as Arthur C. Clarke said.
Thank you for that, RJ. And this is indeed my kill room. I always kill people when there's blue lighting. Just kidding, YouTube. Okay, thanks for your super chat. It's always green lighting. Fractal Escape, thanks for your super chat, said, would either of the debaters comment on the tactic of berating and insulting the other debater as a tactic? Also, both of you were top notch. Thank you. Yeah, what do you guys think of this? The tactic of berating.
Ad hominems are a waste of time on there. I may regard Standing as somebody who is too credulous with his sources, but he's honest about what he believes. I don't for a moment think that Standing is being disingenuous in saying the things that he actually believes are true. And I certainly know that I never say anything that I don't believe is true.
Yeah, I agree. I think ad hominems are a waste of time. I generally try and avoid them. Like I've said it before, I enjoy talking with RJ, and I think he's probably one of the more knowledgeable in this field. So that's why I enjoy talking to him and discussing the tough questions. So yeah, I got nothing bad to say. I enjoyed this debate. Gotcha.
And thanks for your super chat from Fractal Escape, who says, would either of the debaters comment on the... It's embarrassing. We read that. Philip, thanks for your super chat. Says, Standing for Truth, when did the Ice Ages occur in your model?
The Ice Age would have occurred after the flood. I think the mechanisms and the after effects of a global flood would actually be a better explanation for something like the Ice Age, which actually would have revealed land bridges and migration patterns would be easy to explain with the post-flood Ice Age. So yeah, I mean, it's an easy question. Short answer, after the flood.
Yeah, and it's not an easy question for them to deal with. But yes, it is a dogmatic position of Young Earth Creationists that there was just one Ice Age and that it took place post-flood. But the amount of information on not only Ice Ages in the Northern Hemisphere, but ones in India and in Antarctica and various other places that over time, South America, that date back into long different periods and are distributed. I will just be delighted if I can ever see creationists come up with a paleogeography map showing where this Ice Age could account for all of those rocks and yet fit in with all the other stuff. None of them have done it so far.
Gotcha.
Well, I think it's I think it's kind of cool that based on a post-flood ice age that many or nearly all the like, for example, the ice caps, if they were larger and then the oceans were lower, even by, I don't know, 300 or 400 feet, nearly everything becomes connected. So according to our model, migration is easy in a world such as this. There'd be land bridges 1,000 miles
Where
wide.
was India when it had its ice age?
Well, I mean, according to catastrophic plate tectonics, how do you explain India? It'd be the same thing, but it'd be rapid plate movement. CPO.
But where? You realize the ice age is in the southern tip of India, going from the equator northward in its present location.
Right. Are you trying to say that according to at least that continent on the planet, the land bridges being 1,000 miles wide wouldn't?
Oh gosh, no land bridge. India used to be down in the southern hemisphere, parked next to Antarctica and Australia. It moved northward after the Silurian period. That's when it had its ice age. There was a big global ice age with a southern hemisphere polar continent down there, and then it moved forward, slammed into the hot spot where New Zealand is today. That bent it northwards, and it was crossing the Reunion Island hotspots in the middle of the Indian Ocean at the time of the Deccan traps with the dinosaur period, and then eventually slammed into all of this took 100 million years or more, and we can track that in great detail. I have yet to see any creationist account for the paleogeography of their little funhouse world in any level of consistency. Chadwick can't even figure out what Pangea looked like.
Well, I mean, that's the assumed evolutionary deep time history, according to
no. This is their model.
I've got to give.
They've never assembled a workable flood model for any of this.
Because the question was for Standing, I got to give him the last word right now, and go ahead, Standing for Truth.
Yeah, that just assumes snail-paced tectonic movements, uniformitarian processes. For example, we look to catastrophic plate tectonics, meters per second plate movements, and even that's made testable predictions. Some that
and it's wrong.
Confirmed. Well, I mean, the cold slabs that you love so much, they've come true.
Next,
the cold slabs.
I knew that.
Either.
I knew that. We'll talk about that next time.
It's going to be in book 2.
Yes, we definitely can have you guys on again, or if it's on one of your guys' channels or another channel, let us know. We'll tweet it out, and that way people can watch it.
We don't have trouble filling up an hour or 2 or 3, do we?
That's true.
No, sorry.
And want to also say, Casey Jones, thanks for your subscribing today. We're excited to have you with us. I think I missed one earlier. I saw one in the notification that someone else subscribed as well. So I want to say we hope you feel welcome here, folks, no matter what walk of life you're from. And we are excited as, by the way, I just heard we're going to have another, should be a debate tomorrow night as well on secret. It's a different one. We've never hosted, that's for sure. So want to say thanks to our guests. It's been a pleasure to have you, RJ and Standing for Truth. Thanks so much for hanging out with us tonight. We really appreciate it.
And don't accept any wooden penguins, and everybody stay safe.
Everybody stay safe. And James, thanks again. Once again, congrats on the 20K. Like and subscribe. I enjoyed this debate, so we'll do it again someday.
Thanks for that. Appreciate it, guys. And thanks, everybody, for your questions and hanging out with us. We hope you have a great night. Keep sifting out the reasonable from the unreasonable. Take care.